So, reproductive cloning just refers to the process of creating an organism that is genetically identical to the donor organism. And the goal of reproductive cloning is to produce an individual that is exactly like the original in terms of genetic makeup. So, plant cloning has been a long routine practice done for several decades, but animal cloning was once thought to be impossible, particularly with the cloning of adult animals. And that was until March 1997 when we had the birth of Dolly. So, if you've ever heard of a sheep named Dolly, that's what we're talking about now. So, Dolly was the first mammal to be cloned from an adult cell, and her birth really marked a significant milestone in the field of cloning. The process involved transferring the nucleus of an adult cell into an enucleated cell, which is just an egg cell that has had its nucleus removed. So, to prepare the donor cells, they actually starved the cell to help induce a state called G0 or G0. So, we've talked about this previously in the cell cycle. And the G0 or G0 phase is the resting phase where cells do not divide. And these cells, if they're in this stage, they can actually be reprogrammed to initiate development once they are introduced into an enucleated egg, and it's essentially starting the process of cloning. However, Dolly's case really highlighted some unexpected challenges in cloning. So, despite her birth and everything going really well there, Dolly actually had to be put down at just 6 years old. So, this is far earlier than the typical lifespan of a sheep. So, one key factor behind her premature birth were shortened telomeres. So, telomeres are protective caps at the ends of chromosomes, and they shorten every time a cell divides. Because Dolly was cloned from an adult sheep, she inherited older telomeres from the donor cell, and this made her cells age faster than those of the naturally born sheep. So this also resulted in premature signs of aging, which is one of the reasons that Dolly had developed health problems and had to be euthanized early. Dolly's cells seem to retain the memory of the original donor. So even though they had been reprogrammed, some of the molecular marks of the donor cell memory actually remained. And this phenomenon is referred to as cellular memory. So where certain characteristics from the original cell are retained in the cloned animal, and it can lead to unexpected aging and disease-related issues. So this raised questions about the long-term viability of cloned animals and the importance of fully reprogramming donor cells before cloning. So since Dolly's death, scientists have worked to help refine the cloning process and to address the issues, particularly concerning the telomere length and the cellular memory. So cloning still has the potential for agriculture and medical purposes, but Dolly's early death did serve as a reminder for all of the challenges involved in cloning animals. So since then, the cloning of farm animals with desirable traits has become more common. So we have farm animals such as cows and pigs and sheep that are cloned to produce offspring with superior qualities, such as a higher milk production or for disease resistance. So cloning is still being explored for the potential to also help save endangered species by creating clones of animals that are at the high risk for extinction. But despite these advances, the cloning of humans remains prohibited in the United States, and no federal funds can be used to clone humans. There has been success shown in animals. There are still challenges, like I mentioned, including abnormal aging in some of the cloned animals. And these animals can also experience the premature aging, which suggests that we still have a lot of unresolved issues in the cloning process. So additional research, or what they're working on now, is trying to understand and address these challenges fully. So therapeutic cloning is aimed at producing cells, tissues, or organs for medical purposes rather than for creating a whole organism. So the main goal of therapeutic cloning is to generate mature cells that can be used to study cell specialization and develop treatments for human illnesses. So this type of cloning is seen as a really important tool in regenerative medicine and could be potentially used to treat conditions like spinal cord injuries, heart diseases, and even diabetes. So therapeutic cloning can be carried out in several different ways, one of which involves the use of an embryonic stem cell. So these cells are considered a really valuable resource because they're pluripotent, meaning that they can develop into many different types of cells. However, the use of embryonic stem cells does raise ethical questions as they are typically derived from human embryos. So this has led to debates about morality of using embryonic stem cells for research and therapy. Another approach involves the use of adult stem cells, which are found in various tissues throughout the body. So adult stem cells are more limited than embryonic stem cells in the type of cells that they can actually become, but they're still valuable in medical research. So there is ongoing research to overcome the limitations of adult stem cells, like I just mentioned, and also such as their restricted differentiation potential. So therapeutic cloning is potentially really important in the future because it does hold promise for something that we call personalized medicine, where the stem cells derived from a patient's own body could actually be used to generate tissues or organs for transplantation, helping reduce the risk of rejection. But again, we're still facing challenges of, do we have any ethical issues? What are the regulations on therapeutic cloning? And how expensive are these costs going to be in the clinical setting? So genomics is the study of genomes, which is the complete set of genetic material or DNA that an organism has. And the field of genomics explores both coding and non-coding DNA, and it helps scientists understand how genes and the regulatory elements function together. So the Human Genome Project was a major milestone in genomics, and it was completed in 2003 after a 13-year effort to map the entire genome. The project revealed that approximately 3 billion base pairs of DNA are in our human genome, and they identified the genes responsible for human traits and diseases. So one of the key findings of the Human Genome Project was that there is significant variation in human DNA. So for example, some individuals have extra copies of certain genes, which could affect traits such as disease susceptibility. And this discovery really helped introduce or suggest a way for personalized medicine where genetic variations can be used to kind of tailor treatments to individual patients. So interestingly, humans share over 90% of their genes with chimps, which is a clue to our shared evolutionary origins. Humans share also a surprising number of genes with more distantly related organisms, including bacteria. So these comparisons have helped scientists understand both the evolutionary history of different species and the fundamental biological processes that are common across life. So for example, comparing chromosome 22 of humans and chimps has provided insights into how certain genes are related to speech, hearing, and smell, and how these have evolved, and how they possibly play a role in the development of human traits. So while genomics focuses on studying the genetic material of organisms, proteomics is the study of proteins that are produced by these genes. And it's going to explore how proteins are structured, how they function, and how they interact to produce the traits and behaviors of an organism. So since proteins are the key functional molecules in cell, understanding proteomics is a really important field, kind of like especially for drug development and disease treatment. So by studying how these proteins interact with the body, scientists can identify potential targets for new medications and therapies. Bioinformatics is a fairly new field that applies to computer technologies to study the genomes and proteomes. So we have huge amounts of data that are generated from genomic and proteomic research, and bioinformatics really plays a huge role in analyzing and interpreting all of this data that's being brought in. So it does use sophisticated algorithms, and researchers try to find patterns within all of this information. They also try to predict gene function and identify biomarkers for diseases. So bioinformatics can be useful in understanding complex diseases like cancer, where we have multiple genes and proteins that are being involved.